Welcome to the deep dive. This is where we take cutting-edge research and, well, get straight to the core insights quickly. Today, we're digging into one specific, very relevant study. It's got potentially huge implications for criminal justice and finding missing persons. The paper is from the Journal of Forensic Sciences, titled Assessing the Use of Environmental DNA, eDNA, as a tool in the detection of human DNA in water. It's led by Antony Dass and her colleagues, published back in 2022.
Yeah, and our mission here is basically to see if forensic science can effectively borrow this high-tech tool that ecologists have been using for years. You know, environmental DNA, eDNA, it's used non-invasively all the time to track fish or invasive species. But the big question for forensics is, can we actually use it reliably to detect human DNA in places like lakes or rivers?
And the stakes are pretty high, right? I mean, if law enforcement could just take a water sample and get a reliable timeline, maybe how recently someone was there, or even find a trace, that could completely change how searches for remains happen. This study is really trying to pin down that crucial window. How long does this stuff actually last? Okay, let's unpack that term first. eDNA. So eDNA, it's fundamentally simple, isn't it? It's just the genetic bits and pieces that all organisms shed constantly. Things like sink cells, could be urine, saliva, feces, stuff that just ends up in the environment. The real kicker is you don't need the actual organism. You just need the water or soil or even air it was in.
Exactly. And while forensic labs are obviously very experienced with molecular tools on direct samples, pulling usable human DNA out of, say, river water,
Right.
that's a newer challenge. It's promising, but tricky. So for this to ever stand up, maybe in court, the researchers needed to answer that basic question. What's the decay rate? You know, how fast does this DNA signature fade? Understanding that gives law enforcement a potential time frame. There's a person here hours ago, days ago. That's what this paper really aimed to establish.
Makes sense, because as soon as that genetic material hits the water, the clock starts ticking.
Oh, absolutely.
Yeah.
The DNA stability gets attacked by, well, everything. Sunlight, UV light, temperature fluctuations, salt content, and crucially, bacteria and enzymes in the water nucleuses that basically chew up DNA.
Right, which explains why they set up such a controlled experiment to figure out these decay rates. They tracked it over, what was it, 860 hours?
About 35 days, a good long stretch.
And the setup itself, they used these 20-liter plastic chambers, kept things pretty stable, 19 degrees Celsius, a regular 12-hour light-dark cycle, and they aerated it too, so no weird oxygen-deprived conditions. And for the actual human DNA source, just a tiny amount, 100 microliters of whole blood from a single donor.
That's right. And then they compared two scenarios, kind of a clean lab versus the messy real world. So 4 containers had pure distilled water, basically sterile. That was their baseline control.
Okay.
And the other 4 had environmental water. They actually got this from a dam, one chosen specifically because it likely had low background human DNA already in it. They wanted to really isolate the degradation factors in natural water.
And they weren't just looking for any DNA, right? They had 2 specific targets.
Correct. A dual approach, first mitochondrial DNA, mtDNA. You can think of that as the is anyone there signal. It's super abundant in cells, thousands of copies. So they used a really sensitive method, qPCR, just to detect its presence and measure how much was there.
And the second target.
That was nuclear DNA, nDNA. This is the who are you signal. For this, they used STR profiling, short tandem repeats. That's a standard forensic technique that generates a unique genetic fingerprint to potentially identify an individual.
Okay, so detection versus identification. Got it. Now, you mentioned the messy real-world aspect, and didn't they run into a problem right there with the water itself during filtering?
Yes. That was a key methodological point. The environmental water, because it had sediment, algae, just general gunk.
Yeah.
They could only manage to filter about 50 milliliters through the fine 0.22 micrometer filters they were using.
Only 50 milliliters, but for the clean distilled water.
They could filter 500 milliliters, 10 times the volume.
Wow. Okay, hold on. Doesn't filtering 10 times less volume from the environmental sample potentially skew things? I mean, if you start with less water, aren't you just less likely to find trace amounts regardless of the decay rate? Does that mean the real-world decay might actually be worse than reported?
That's a really sharp point. And the paper does acknowledge this challenge. It doesn't necessarily bias the rate of decay calculation itself that's about the proportional loss over time, but it absolutely highlights a practical difficulty in the field. It's harder to find that needle if your haystack is smaller and full of debris. Right. It shows collecting enough material in the first place from natural water is a major hurdle. You just have less opportunity.
Right. Okay. That makes sense.
So let's get to the findings then, because this is where that difference between the lab water and the dam water really shows up, isn't it? The DNA decline was exponential, sure, but the speeds were, well, Vastly different.
Oh, dramatically different.
The environmental water acted like a DNA shredder. The decay constant they calculated, K, was 0.070 per hour. That translates to a half-life of only 9.86 hours.
Under 10 hours for half of it to be gone.
Exactly. Half the detectable human DNA material destroyed in less than a working day.
And the distilled water.
Much slower decay. The constant there was 0.019 per hour, giving a half-life of 36.71 hours. So nearly 4 times more stable in the clean water.
Wow.
It tells you pretty conclusively that the biological and chemical components in natural, untreated water are the biggest enemies of this type of evidence.
Okay, so let's talk persistence windows. For that basic detection signal, the mtDNA, the "are you there" bit, what were the maximum times?
Well, there's some hope there. In the environmental water, they could still detect human mtDNA for up to 11 days.
11 days. That's actually longer than I might have guessed given the fast decay.
It is significant. But compare that to the distilled water where it lasted the entire experiment. 35 days. Still detectable at 840 hours.
So 11 days is potentially useful for narrowing down a search time frame, pushing it from hours to almost 2 weeks. But you mentioned that rapid initial decay.
Right, the speed is brutal initially in natural water. Within the first 24 hours, the concentration dropped by 90%.
90% gone in a day.
And after 48 hours, 96% was gone. So while it persists at low levels for 11 days, most of it vanishes incredibly quickly.
And what did this rapid decay mean for the nDNA, the identification part, the STR profiles?
That's where it gets really challenging for forensic identification. Full STR profiles, getting all 42 alleles, the complete genetic fingerprint needed for a solid ID. They only got those right at the very beginning, time 0, in both water types.
Only at time 0, so basically instantly degrading from an identification perspective.
Pretty much. In the environmental water, the number of alleles they could detect just dropped off progressively. And the key finding, after just 24 hours, they couldn't detect any alleles. 0.
24 hours, that's incredibly fast. So thinking practically, if something happens, say, overnight, by the time investigators might realistically get a sample the next day, the chance of getting a unique ID from the water itself is basically gone.
In natural water, based on the study, yes.
Yeah.
The window for identification seems vanishingly small. In the distilled water control, they did get partial profiles, sometimes just 1 or 2 markers much later, even up to 840 hours. But partial isn't usually enough for a court ID.
And they also saw a big difference in just the signal strength, didn't they? Something about inhibition.
Absolutely. That filtering issue we talked about likely plays into this too. When they measured the peak heights, the RFUs, which indicate signal strength, at time 0 in distilled water, the average peak height was huge, over 91,000 RFUs.
Okay.
But in the environmental water, at the same time point, it was only about 2,500 RFU, a massive reduction.
And that suggests?
It strongly suggests inhibition. Stuff in the environmental water, probably things like humic acids from decaying plant matter, is interfering with the PCR process itself. It's like trying to copy a faint message with sticky tape gumming up your photocopier. It'd prevent accurate amplification.
Okay, let's zoom out then. We have this mtDNA detection window of up to 11 days, but an nDNA identification window of less than 24 hours in real-world water. What's the bottom line for law enforcement? What can they actually do with this?
Well, the big takeaway is that sampling for human eDNA in water is a potentially viable tool, but primarily for detection within that, say, 11-day window. It might not tell you who it was, but it could strongly indicate that human remains or biological material were present in a specific area recently. That's huge for focusing search efforts, saving time and resources.
So narrowing the search area.
Exactly. And even those very weak partial STR profiles, like the 3 alleles they found at 24 hours in the environmental water, they aren't useless. They probably wouldn't identify someone definitively, but they could potentially be used for exclusion, ruling people out, or maybe suggesting a possible link that justifies bringing someone in for comparison or focusing the investigation further. It's still valuable intelligence.
So the message seems to be focus on the mtDNA for detection because it's more abundant and lasts longer.
That seems to be the practical advice, yes. It's just structurally tougher, and there's so much more of it per cell compared to the single-copy nuclear DNA. That gives you the best chance for that longer detection window. The nDNA, while ideal for ID, is just too fragile in that environment.
We should also probably touch on contamination, right? Because that seemed like a real issue even in their controls.
A huge issue and a critical real-world challenge. Yes, they found background mtDNA contamination even in their highly controlled setups. And interestingly, in the distilled water control at time 0, they got a partial STR profile from a male that didn't match the female blood donor.
Wow, even in the pure water.
Yeah, it just highlights that trace human DNA can be almost anywhere, maybe from lab reagents, handling, aerosols. It means forensic protocols using eDNA need incredibly stringent controls and careful interpretation to avoid false positives or misinterpretations. Background noise is a real factor.
Okay, let's talk limitations. We've hit on the PCR inhibition from things like humic acids and that difficulty filtering enough environmental water. You mentioned they suggested some ways around the filtering problem.
They did. Since filtering small volumes limits how much trace DNA you can capture, they suggested exploring things like pre-filtering steps, maybe using a filter with larger pores first to get rid of the big debris, then filtering the remaining water through the fine filter. Or perhaps just using slightly larger pore filters overall. It's a trade-off, though. You might remove inhibitors, but also risk losing some DNA trapped in the larger particles you filter out. It needs more work.
Makes sense. And what about the source material? They use blood here.
Right, that's another limitation. This study used fresh whole blood. But in a real case, you might be dealing with saliva, semen, bone fragments, decomposing tissue, all sorts of things. Each of those might shed DNA differently and have its own unique decay profile in water. So we need more research on various tissue types to build a better reference library for how different biological materials behave as eDNA sources.
Which leads nicely into future research. What else needs figuring out?
Well, a big one is dynamic flow. This study used static tanks. But what happens in a river? How far does the eDNA travel downstream? Understanding transport distance is absolutely crucial for defining a realistic search radius from where a sample is taken.
Yeah, that seems critical.
And the other key area is mapping background levels. We need to understand the normal amount of human eDNA noise in different water systems. Think about wastewater outlets, popular swimming areas. These places will have a lot of background human DNA. Before you can confidently say the detection is linked to a specific forensic event, you need to know what the baseline looks like in that environment.
So despite the challenges, this research does seem to provide real value. It confirms eDNA sampling can be a cost-effective addition to the toolkit, especially for that initial detection within roughly 11 days using mtDNA.
Absolutely. It offers a concrete time frame for detection, which is incredibly useful. And maybe that leads us to a final thought for you, the listener, to consider. Given that this study found background human DNA even in controlled settings, and knowing that many natural water sources near human activity likely contain a mix of DNA from swimming, wastewater, runoff, how essential is it for forensic teams to first create detailed baseline maps of the normal human eDNA levels in their local waterways? Can they really interpret a positive find confidently without first understanding the background noise? How do you find that one critical signal if you don't know what the everyday chatter sounds like? Something to think about.